Hello everyone, I am Dr. Sirajul Akfazi. I welcome you in my YouTube channel. Today I am going to talk about enzyme and I hope this video will be helpful to you. So friend, let us to start enzymes. Enzymes are biological catalysts, you all know that, which is synthesized by a living cell and that accelerate the biochemical reactions. The orderly course of metabolic process is only possible because each cell is equipped with its own genetically determined set of enzymes. It is only this that allows coordinated sequence of reactions, metabolic path involved in many regulatory mechanisms, and almost all enzymes are proteins except catalytically active ribonucleic acid, the ribozymes. Friend, enzymes are characterized by three distinct features: catalytic power, ability to catalyze biochemical reactions, accelerating reactions rate as much as 10 to the power 6 over uncatalyzed levels, far greater than any synthetic catalyst. As specificity, a given enzyme is very selective, both in the substances with which it interacts and in the reaction that it catalyzes. Regulations, metabolic inhibitors, and activators. Friends, enzyme nomenclatures. Very important. Traditionally, enzymes often were named by adding the suffix "-ac" to the substrate upon which they acted. Example: phosphatase, urease, catalase, proteases. Confusion arose from these trivial names. So a new system of nomenclature of enzyme was developed based on the nature of reaction it helps. 6 classes of reactions are recognized. Within each class are subclasses, and under each subclasses are subclasses within which individual enzymes are listed. Friends, enzymes are classified on the basis of action it performs: oxidoreductases, oxidation reduction reactions; phosphate dehydrogenase; transferases, transfer of functional group; methyl transferases, carboxy transferases; hydrolysis reactions, carboxylic ester hydrolysis; isomerases, isomerization reactions; epimerases, liases, addition to double bonds, carboxylases, aldehyde liases; ligases, formation of bond with ATP cleavage, amino acid, RNA ligases. So friend, intracellular and extracellular enzymes. Intracellular enzymes are synthesized and retained in cell for the use of cell itself. They are found in the cytoplasm, nucleus, mitochondria, and chloroplast. Oxidoreductase catalyzes biological oxidation enzyme involved in reductions in mitochondria. Intracellular, extracellular enzymes are synthesized in cell but secreted from the cell to work externally. Digestive enzymes produced by the pancreas are not used by the cell in the pancreas but are transported to the chemical properties. Most of the enzymes carry out their functions, relying solely on their protein structure. Many other requires non-protein components cofactor, usually metal ion or non-proteins organic part coenzyme. Less complex than proteins, tend to be stable to heat. Many coenzymes are vitamins or contains vitamins as a part of their structure. Functional unit of enzyme is known as holoenzymes, apoenzyme plus coenzyme. Apoenzyme, what is apoenzyme? Protein without any catalytic activity. Now friend, if enzyme is made of single polypeptide monomeric enzyme, ribonuclease or trypsin. If the enzyme is made up of more than one polypeptide oligomeric enzyme, and then example lactate, dehydrogenase, aspartate, transcarbamylase. Multi-enzyme complex have multiple enzymes you need to carry out different reactions in sequence. So friend, cofactor and enzyme, coenzyme, thioamine polyphosphate, pyruvate dihydrogenase, flavin adenine nucleotides, monoamine oxidase, nicotinamide adenine dinucleotide, lactate dehydrogenase, pyridoxal phosphate, glycogen phosphorylase, coenzyme A, acetyl coenzyme carboxylase, biotin pyruvate carboxylase, 5-deoxy adenosyl carbylamine, methyl malonyl mutase, and tetrahydrofolate thymidylate synthesis. Metal zinc 2 plus, carbonic anhydrase, zinc 2 plus, carboxypeptidase, magnesium 2 plus, eco RB, magnesium 2 plus, hexokinase, nickel 2 plus, urease, molybdenum nitrate reductase, selenium glutathione peroxidase, and like that. Now friends, enzyme kinetics. The quantitative measurement of the rates of enzyme catalyzed reactions and the systematic study of factors and that affect these rates helps in the analysis, diagnosis and treatment of the enzyme imbalance that underlie numerous human diseases. The levels of particular enzymes serve as the clinic indicator for the pathology. Myocardial infarctions, prostate cancer and damage to the liver. Enzyme kinetics. Friends, any biochemical reaction constitute A plus B gives C plus D, where A, B are the substrate and C plus D are product. A study of enzyme kinetics has two components, that is Gibbs free energy, directions and equilibrium state of substrate and product, and activation energy, mechanism of reaction and rate of reactions. Now friend, what is the Gibbs free energy change? Delta G, also called either free energy or Gibbs energy. It describes the quantitative from both the direction in which the chemical reactions will tend to proceed and the concentration of the substrate and the product that will be present at the equilibrium. Mathematically, you know del G is equal to del Gp minus del Gs. Del Gp is sum of the free energy of formation of the reactions product and del Gs sum of the free energy formation of the substance. The sign and the magnitude of the free energy change determine how far the reaction will proceed, and if delta G is negative, then the reaction proceeds in forward direction spontaneously. So friend, what is the activation energy? Any reaction doesn't proceed directly to the product formation. There is always a transition state between the ground state and products. Activation energy, the difference between the energy level of the ground state and the transition state. The functions of the catalyst is to increase the rate of reaction. It does not affect the reaction equilibria. So enzyme just lower the activation energy. So friends, enzymes free energy reactions coordinate ground state and P product ground states. You can see the transition state here. So factor affecting the enzyme activity. The contact between the enzymes and substrate is the most essential prerequisite for enzyme activity. The important factors that influence the enzyme reactions are concentration of the substrate, concentration of enzyme, temperature, pH, product concentration, activator, time, light and radiations. So concentration of substrate. The frequency at which molecules collide is directly proportional to their concentration. Rate directly proportional to concentration of A and B. Rate is equal to K, A to the power N, B to the power N, where Na and B give P and K is a rate constant. The sum of the molar ratio of the reactant define the kinetic order of the reaction. In the examples above, reaction is said to be N plus M order overall, but N order with respect to A and M order with respect to B. Now Michaelis-Menten Constant Km, also known as Halden's constant, substrate concentration to produce half maximum velocity in the enzyme catalyst reaction. Km is a constant and characteristic feature of a given enzyme, the strength of enzyme substrate. Low Km values indicate a strong affinity between the enzyme and substrate. Majority of enzyme Km value 10 to the minus 5 to 10 to the minus 2. So you can see the graph, initial velocity and substrate concentration in millimolar, half V max and complete V max. Here the formula V naught is equal to V max concentration of substrate divided by Km plus concentration of S, Michaelis-Menten reactions. So friend, concentration of enzyme. Reaction velocity is directly proportional to the concentration of enzyme. Serum enzyme for diagnosis of disease. known volume of the serum and substrate taken at optimum pH and temperature. Enzyme is assayed in the laboratory. So friend, what is the role of temperature? Velocity of enzyme reaction increases with the increase in temperature up to maximum and then declines. Increase in the temperature causes increase the kinetic energy of molecules. A bell-shaped curve is usually observed. Temperature coefficient increase in the enzyme velocity when the temperature is increased by 10 degrees Celsius. Optimum temperature for most of enzyme -40 to -45 degrees Celsius. Beyond the 50 degrees Celsius there is a denaturation of enzymes. So what is the role of pH? Most intracellular enzymes exhibit optimum activity at pH values between 6 to 8. Balance equation between the enzyme denaturation at a high or low pH and effect on the charge state of the enzyme and the substrate or both. Exception pepsin and acid phosphatase and alkaline phosphatase. You can see here. So activators, certain metallic cation, magnesium, zinc, calcium, cobalt, copper, sodium, potassium. It acts in various ways, combining with the substrate. Formation of ES metal complex, direct participation in the reactions and bringing a conformational change in the enzyme. There are two categories of enzyme requiring metal for their activity. Metal activated enzyme, not tightly held by the enzymes and can be exchanged easily. Friends, the metalloenzymes hold the metal tightly. Alcohol dehydrogenase, carbonic anhydrase, alkaline phosphatase and carboxypeptidase. So other factor, product concentration. The accumulations of the reaction products generally decrease the enzyme velocity. Light and radiation, exposure to UV, beta, gamma and X-ray activated certain enzyme. Formation of peroxidase. UV rays inhibit the salivary amylase activity. So friend, mechanism of enzyme action. Active site. The active site of enzyme is the region that bind a substrate, cofactor and prosthetic groups and contains residue that help to hold the substrate. Active site generally occupy less than 5% of the total surface area of enzyme. Active site has a specific shape due to its tertiary structure of protein. A change in the shape of the protein affects the shape of the active site and function of the enzymes. So friends, what is the active site? You can see the substrate molecule. The substrate molecules are the chemical that an enzyme act on. They are drawn into the cleft of the enzyme. Enzyme molecules, the complexity of the active site is what makes each enzyme so specific. So active site, the active site contains both binding and catalytic region. The substrate is drawn to the enzyme's surface and the substrate molecules are positioned in a way to promote aeration, either joining two molecules together or splitting up a larger one. So friends, active site can be further divided into active site, binding site and catalytic site. It chooses the substrate and bind it to the active site and it performs the catalytic action of enzymes. So mechanism of enzyme actions. The catalytic efficiency of enzyme is explained by the two perspectives, thermodynamic changes and process at the active site. And thermodynamic changes, all chemical reactions have energy barrier between reactants and products. You know that the difference in transition state and substrate is called activation energy barrier. The process at the active site, covalent catalytic, acid base catalysis, catalysis by the proximity, catalysis by strain. The covalent catalysis, the enzyme form covalent linkage with the substrate forming transient enzyme substrate complex with very low activation energy. Enzyme is released unaltered after the completion of the reactions. So acid-base catalysis, mostly undertaken by the oxidoreductase enzyme, mostly at the active site. Histidines in the present which act as the both proton donor and proton acceptor. So friend, catalysis by the proximity. It is catalysis molecules must come in bond forming distance. When enzyme binds, a region of the high substrate concentration is produced at active site, and this will orient substrate molecules especially in the position ideal for them. So friend, catalysis by the bond strain, mostly undertaken by the lysis. The enzyme substrate binding causes reorientation of the structure of site due to its strain condition, and thus transition state is required, and here bond is unstable and eventually broken. In this way, bond between the substrate is broken and converted into products. So friend, lock and key models. The purpose of EMI fission in 1984, lock and key hypothesis assumed the active site of enzyme are rigid in the shape. There is no change in the active site before after a chemical reaction. So friend, hope this video will be helpful to you. If you like this video, then subscribe my channel, and thanks for watching.